Welcome to the AME Food Testing Show. Today's topic, validated real-time reverse transcriptase PCR methods with Manohar Furtado, Ph.D. Manohar is currently consulting for Biology for Global Good. He is or was the lead R&D and molecular diagnostic team leader at Applied Biosystems, Celera Diagnostics, and Life Technologies for over 12 years. He was assistant professor of pathology and director, clinical virology and retrovirology laboratories at Northwestern University Medical School for 7 years. Over 15 years, he worked in life sciences, primarily in academic research and clinical research as a principal investigator at the Robert H. Lurie Comprehensive Cancer Center. His Ph.D., master's, and bachelor degree were obtained from Poona University. His area of expertise is biochemistry in the specific designation of protein structure and evolution. He has over 70 publications in peer-reviewed journals. He also has presented over 200 oral presentations and posters at major and international conferences. He has 2 issued patents and 10 pending patent applications. Now, welcome with me to the AME Food Testing Show, Manohar Furtado. Manohar, can you update us on any of your other activities?
Yeah, so I was with Life Technologies and Applied Biosystems, as you mentioned, for about 12 years, heading up the R&D group there largely for diagnostics in terms of testing for contamination in food, pharmaceuticals, and in animal health as well. I started my own company in December 2012, Biology for Global Good, and I've been consulting for a few biotechnology companies. I was also appointed to the National Biodefense Science Board by Kathleen Sibelius, so it's an advisory panel that works with DHHS to implement some policy decisions. So that's what I've been doing more recently.
Excellent. What methods are traditionally used to detect avian influenza virus infections?
So influenza viruses have been around for a long time, and the oldest detection tests were largely cell culture based. So you would take a sample from someone who's infected, put it on a bed of cells in tissue culture, and then wait for the virus to grow, and then detect it by an antibody or something like that. Typically, those tests used to run for 1 to 10 days. More recently, companies have come up with rapid tests where they have antibodies generated against recombinant influenza proteins, and they use it for direct detection of influenza-related antigens. Those tests are more sensitive and can also give you results at a faster turnaround time, so typically maybe within the day. Ultimately, the most advanced form of detecting influenza viruses is picking up the RNA itself, and that's usually a PCR-based test preceded by a reverse transcription step to convert the RNA into a DNA and then amplify by PCR and detect it. So in a nutshell, that's what's being done in influenza testing.
Very good. What is real-time reverse transcription PCR?
Okay, so, the genomes of organisms are mostly DNA, but there are a large group of pathogens that have ribonucleic acid as the genome. Examples would be HIV, hepatitis C virus, and influenza virus, among the more well-known viruses. So if you want to detect them by using polymerase chain reactions, you have to first convert the RNA form of the genome into a DNA strand. And that reaction is catalyzed by an enzyme called reverse transcriptase, which uses DNA as a template and then make a complementary RNA strand. And once you have the complementary cDNA strand, that strand is then used for amplification by the traditional polymerase chain reactions to generate thousands of molecules, which makes it easy to detect with a probe.
Dr. Furtado, how do RT-PCR technologies compare with culture and other traditional methods?
There's been a lot of study on this with the large number of viruses, as I mentioned, HIV, hepatitis C, and influenza. And typically, the RT-PCR technologies will pick up the virus much earlier, typically maybe 4 to 5 days earlier than what you can see in culture. So it again depends on the viruses. Influenza grows relatively rapidly in cells. When you look at some other viruses, they grow a lot more slower. But in general, the culture techniques are slow because you have to grow the virus to a certain level before you can actually detect it by looking at what's called a cytopathic effects or by looking at staining with antibody. In case of the RNA, it's detectable earlier, and because the PCR techniques are so sensitive that they can detect in the 5 to 10 molecule range, they become it becomes a lot more faster to detect these things and much earlier than get a much earlier result than what you'd get with traditional culture methods.
What are the limitations of liquid RT-PCR assays?
Well, RT-PCR assays in liquid form, do work very well, but some of the limitations would be in how much sample can you put into the reactions. So that's one of the things. The other aspect of this is stability. So you have to make sure it's stored at -20 or -70 or some, cold storage place to ensure that the reactions work down the road. So for instance, if a company is shipping things out to, let's say, a faraway place, there's transport, there's the kits reaching the lab, they open it, and they want to use it for a few months. So you need to be sure that it is stable in liquid form for the period of time that people are going to use it.
Very good. Your research has particularly focused on a technology called lyophilization. Can you explain that in layman's terms for our audience of 79,000 food production, food quality, food safety, and food security managers?
Yeah, so lyophilization is a process by which you essentially take the water out of the reaction. So all biological reactions, if they need to proceed, require water. And then when you lyophilize, essentially what you're doing is reducing the water content to a level where chemical reactions are not supported. Now, this helps in a couple of ways. One is the enzymes and oligonucleotides and the NTPs and material that typically form a liquid PCR reaction can now be in a single tube and remain inactive as long as you need to. So when you ship the assay around and it goes to labs, you can typically store this at room temperature or at 4 degrees centigrade, and it's stable for long periods of time. That's one advantage. The second advantage is because of the low water content, you don't get contaminating bacteria or other organisms growing within the reaction itself. So there are definite advantages in terms of stability and ease of use when it comes to using lyophilized reactions. The other thing I should mention is the ease of use. Because you have all the components lyophilized in one pellet, you don't need to mix 3 or 4 reagents when you set up a reaction, and that's typically useful in a routine testing lab. The last thing I'd like to say is because of the lyophilization, you can now add a larger volume of your sample. So typically, if you're setting up a 50 microliter reaction, In the liquid format, you may be able to accommodate somewhere between 15 to 20 microliters of the sample. In a lyophilized reaction, you can put in as much as 50 microliters. So there's a big difference in terms of how much material goes into the reaction to be tested, and that helps with sensitivity.
Well, Dr. Furtado, you recently participated in a fascinating study on the assay trials of a RT-PCR lyophilized test. Can you review that for our audience?
Yeah, so we have been when I was at Life Technologies, we were generating lyophilized tests for detection of a large number of food pathogens and also for influenza. So one of the key testing trials we did was in collaboration with the VLA, specifically Marek Slomka's group in the UK, where we send them lyophilized avian influenza assays, that is, assays that detect H5N1. Internally, we had done detections to show that we can detect anywhere between 4 to 16 molecules with the lyophilized format compared to requiring 16 to 80 molecules for the corresponding red assays, which means that we had roughly a 4-fold gain in sensitivity. This was internal work, so to test it out, we sent assays to the VLA, and they looked at testing actual influenza samples and compared the red assay versus the lyophilized assay. We used about 19 influenza virus panels. And the general result of this is that the wet samples detected only 86.3% of the total collection. That is, 38 out of the 44 samples were detected by the wet assays, and all 44 were detected by the lyophilized assays, indicating a higher degree of sensitivity. And I believe a large amount of this comes in play because of the larger amount of samples that you're able to use.
Excellent. So if I could summarize your trial result at 100% sensitivity specificity for the panels for avian influenza. By the way, in California, where I believe you live as well, there are a growing number of cases for this challenge, isn't there?
Yeah. So the detection limits and sensitivity of the assay also, to be fair, depend on the design of the primers and the probes. And then, flu is a continually changing viral sequence format. So there's the evolution of virus. Each year you find different strains around. So obviously you have to know sequence information about these strains to design effective assays. Given that, just strictly comparing for a given target, a wet assay versus a lyophilized assay, we found greater sensitivity with the lyophilized formats. And you're right, in California there's always challenges with flu worldwide, but in California as well.
Well, Dr. Furtado, I'd like to now allow you to brief our audience on your consulting practice and what kind of clients you accept, and any other information you'd like to share with our group.
Okay, so I've been generally interested in healthcare science and healthcare research at Northwestern. I worked on virology largely, joined the company in 2000, put out an HIV genotyping kit that was FDA approved for detecting mutations in HIV, and then built up business units that did food pathogen testing for contamination in pharmaceuticals. Like the drug industry requires a lot of testing before they release materials out. We built tests for human pathogens, animal pathogens. So generally, I'm interested in health and safety applications where you can use molecular diagnostic tests to make sure, for instance, the products that you put out do not contain any adulterant or pathogen. I'm also interested in developing similar assays for the clinical diagnostic market, where you do testing to see if people are infected with pathogens. So that's been the general strategy that I've been using. I've been consulting for a couple of companies to help them develop these assays and then move them into the clinical space in two different areas, largely infectious disease, and the second area of application is cancer diagnostics, more specifically and more recently, a group of tests called companion diagnostics, where you use the test to predict the efficacy of a drug. So that's largely what I'm focusing on as far as my clinical practice goes. As far as my industry practice goes, I'm generally a molecular biologist with expertise in infectious disease and cancer. And I try to apply that to solutions that biotechnology companies need in helping them develop assays, move it into manufacturing, and then take it through for appropriate approval, either, let's say, with FDA or the AOAC or AFNOR or ICH, depending on the particular area they're working in. And that's generally been my focus. Thank you.
Well, Dr. Furtado, I'd like to thank you for your volunteering to brief our audience on this growing, effective technology, and allow you any final comments prior to the closure of this interview.
Well, it's been a pleasure talking to you guys, and like I usually try to tell people within the food industry and pharmaceuticals is your tests are as good as the knowledge that you have about the pathogen. So I just see this in general as a twofold approach. One is you need a large amount of research to sequence new pathogens and define the variability in the pathogens across the globe, because we're living in a global community now. A contamination in one place in Australia could end up in some other plant here in the U.S. So the global knowledge database of pathogen sequences is important, and then use that to detect highly sensitive assays of detection. So that's all I have to say.
Thank you very much, and we wish you a great day.
Thanks, Andy. Bye-bye.
Bye now.